Good afternoon, everyone. Welcome to Webinar Series 2.0, based on the topic Biotechnology and Bioinformatics in Health and Medicine. Myself, Dr. Rachana Banerjee, working as an assistant professor in JIS IASR, Center for Health Science and Technology. I feel myself privileged to introduce today's speaker, Professor Sujoy Chattopadhyay. Professor Chattopadhyay is the Associate Professor in the Center for Health Science and Technology, JIS IASR. He did his postdoctoral studies and research professorship in the Department of Microbiology, University of Washington, Seattle, USA. His area of research is computational and evolutionary genomics of microbes with special interest in microbial virulence and antibiotic resistance. Dr. Chattopadhyay is an outstanding researcher with excellent publication list in reputed journals like Nature Group and National Academy of Sciences. Today, he is going to deliver us an exciting talk on microbial evolution, infectious diseases, and computational genomics. Hope all of us will enjoy his talk. Let's join his talk. And if you have, after his talk, if you have any query, please write in the question and answer section. He will be happy to answer you all the questions after his talk. Let's listen to him.
Thank you so much, Dr. Banerjee, for this wonderful introduction of me. Hope you can hear me, right?
Yes, we can hear.
Yeah, okay. Good afternoon, everyone. Good afternoon to all the attendees who are attending our talks, like this series of presentation we are doing on biotechnology and bioinformatics in the field of health and medicine and research and career prospects. We have talked about it. We have talked about our institutes. Today, I believe I can share the screen. Let me share. Is the screen visible, Dr. Banerjee? Dr. Banerjee, is the screen visible to you?
Yes, it's visible.
Okay. Okay, so you all know, whoever is attending this presentation, all of you know the title of the presentation, and although it's intended for the students primarily of bachelor's and master's level, and maybe someone who are willing to do some research work in the field of infectious diseases or in the field of bioinformatics research, might be interested in this presentation. But at the same time, I consider that many of you who are attending, many of you are young enough to kind of to be not quite aware of all the terms that are there in the presentation. So as it says, microbial evolution, infectious diseases, and computational genomics. So we'll talk about this, don't worry. I don't know what's happening here. It's okay. So there's a first slide. And before we go, I believe for each and every presentation you have heard so far in this series of talks organized by Center for Health Science and Technology of our institute, JIS Institute of Health Studies and Research, Kolkata. We have mentioned about our infrastructural facilities. We have mentioned about our academic programs, about the prospects you can have after completing your master's work. So This time, I actually, many of you are attending this talk, whoever attended the last few talks. So just not to bore you enough, here I'm going to go a little different. I'm going to show you a video that will talk everything about this thing.
A vision emerges in the mastermind of Padmasri Professor Ajay Kumar Ray to develop a unique postgraduate research institute with a difference, promoting an interdisciplinary approach, offering translational research-based education, and focusing on areas with high quality. A vision emerges in the mastermind of Padmasri Professor. A vision emerges in the mastermind of Padmasri Professor Ajay Kumar Ray to develop a unique postgraduate research institute with a difference, promoting an interdisciplinary approach, offering translational research-based education, and focusing on areas with high-quality job prospects and entrepreneurial opportunities. Fortunately, JIS Group educational initiatives joined hands in this dream project, and then, in mid-2019, under the aegis of JIS University.
I don't know, there is some problem going on there. I don't know what's going on. I'm trying to show the video and it's stopping somewhere in the middle.
An interdisciplinary approach. A vision emerges.
I don't know how I can control this.
A vision emerges. Postgraduate Research Institute with a difference, promoting an interdisciplinary approach. A vision emerges in the mastermind of what joined hands in this dream project, and then, in mid-2019, under the aegis of JIS University, JIS Institute of Advanced.
I'm extremely sorry. I'm not sure what's going on here. Suddenly, it's going back and forth.
Especially for direct medical applications.
Let's move forward a little bit, and let's see if the problem still persists.
Academic and industry sectors of health science can only be ever increasing. On one hand, there's a continuous need for biotechnological development, especially for direct medical applications. Simultaneously, the advent of newer high-throughput technologies a vision emerges. This scenario puts qualified bioinformatics professionals, especially the ones with expertise of analyzing medical biotechnology data, are in high demand for top salary jobs worldwide. Keeping this in mind, CHEST offers 2-year MSc in medical biotechnology and bioinformatics, 2-year MTech in bioinformatics, and regular as well as integrated master's PhD program in related fields. But why would you select CHEST? Here are 3 reasons why CHEST is the best nest to hatch your career flight in health and medicine. Number 1, state-of-the-art molecular genomics, biotechnology, and bioinformatics laboratory with avant-garde equipment like PCR, real-time PCR, tape station, gel documentation system, CO2 incubator, fluorescence and UV spectrometer, biosafety cabinet, fume hood, and many more, along with ample laboratory workspace. And finally, last but not least, Illuminamycin, one of the highest ranked platforms for gene. A vision emerges, be the only private educational institute and one of the very few among all public plus private institutes in Eastern India to have the in-house facility of this high-throughput next-generation sequencing and data analysis. Number 2, internationally experienced faculty team with prestigious research grants, top-tier journal publications, and global collaborations. Multifaceted domain knowledge of CHEST faculty. A vision emerges. This aims a paradigm shift in infectious diseases research in India. The expertise in the CHEST faculty team spans from molecular microbiology and post-pathogen interaction, adaptation strategies of microbial pathogens, evolution of microbial virulence and antibiotic resistance, molecular modeling to explore functional insights of biomolecules. A vision emerges. Nature Life Science and Applications, Nature Protocols, Nature Communications, Journal of the American Chemical Society, plus Computational Biology, Proceedings of the National Academy of Sciences USA, to name a few. This has been possible through worldwide collaborations with academia and industry, and prestigious research grant funding from the Government of India, and welcome. A vision emerges. Our biotechnology and bioinformatics programs in health and medicine. Along with a solid foundation of different aspects of medical biotechnology, computational biology, and bioinformatics applications, our MSc of vision informatics, systems biology and biological networks, big data in bioinformatics, and so on. And expertise in any of these fields can readily offer lucrative jobs in pharmaceutical and biotechnology industries, in hospitals and clinics, in bioinformatics data analysis companies. A vision programs consist of topics like research methodology, career development in entrepreneurship, and a vision motivates students to develop an entrepreneurial mindset. So, why delay? Join CHESS, the best net to hatch your career flight in health and medicine, and target the top together.
Okay, finally, we could get through the video somehow. There were many disturbances, many problems that we faced, but somehow I believe you could get a glimpse of what we can offer as part of our academic programs, as part of our research, as part of our infrastructural facilities and faculty team strength. A couple of additional points that you might have missed in this presentation is that I don't know what's going on, why it's moving automatically. Yeah, it's just magic going on here. Okay, so what I have faced it. I mean, many people are coming for interview session to us. I mean, we are having an interactive session through which we offer admission to the interested and motivated candidates. And many of us many of them are explaining to us that, well, we did have bioinformatics program in our bachelor's or maybe in our master's, whoever is coming for MTech program. But in bachelor's and master's, whatever bioinformatics we learned as part of our biotechnology, overall biotechnology program, that is very repulsive. I mean, people say that, well, do we need to know programming language? Do we need to know all these other complicated stuff of statistics and mathematics and all these things, which biologists are normally scared of? The thing is, this is a complete misconception. Definitely, knowing a little bit of statistics, knowing a little bit of programming knowledge would be an added advantage. No question about that. But I have seen very successful bioinformatics researchers all over the world ruling in very top quality US universities there. They're doing research without knowing a bit of computer programming. Believe me, it's all about understanding the biology and how you can use. I mean, there are several computer programs available, bioinformatics programs, software, tools, databases are available in public servers. So you can use them. You don't need to build your own, or even if you need to build your own, you can hire a programmer who can do it for you. But the most basic thing for you to know the biology, that is the most basic thing, because I have seen several bioinformaticians coming from, say, statistics background or some other kind of backgrounds, a computer scientist and maybe electrical engineer. They do a lot of good stuff in developing software tools, but many of them you can hardly use biologically meaningfully. So please, I want to assure you, there is nothing you should be scared of when you come to know about bioinformatics program. Definitely bioinformatics is something that we need for any kind of biological studies nowadays. It's kind of a mandatory thing. And you can see the amount of data is being generated every moment, health data, in data of medicinal research. So you have to tackle them, you have to analyze them, what's going on. I'm just continuously struggling with this mouse thing. What actually is mouse is behaving like I'm the rat and mouse is in my hand. I'm just trying to control and it's not being controlled. Okay. So what I'm talking about, and in many courses, whoever took some courses in, say, undergraduate program, in bioinformatics, people have told some jargons, the faculty members have told some jargons which we couldn't understand. This is what the students who are coming to face our interview are talking about. It's, well, for example, today's talk, you can see it's microbial evolution, infectious diseases, and computational genomics. So it can also be said something like this: computational evolutionary genomics of microbes and infectious diseases. If I want to make it more complex, more complicated, to let people know that, well, I know something that you don't know, just to humiliate someone. So for example, there is one word in English, many of you might be knowing this, floxinosinihilipilification. So this means, this is the longest word, 29-letter word in English dictionary you can find. It means the action or habit of estimating something as worthless or simply nothing. You can say nothing, you can say floxinosinihilipilification. So that is how people kind of nowadays in many classes deal with the students. So that is not what we want to do. We want the students to enjoy the subject, whatever he or she is coming to learn. So that's how that is the motto we have. And another important thing is, even though it's a private institution, definitely private institutions are there to make money. Everyone knows that. But normally, not normally, it's except for this one, I can say 100% of institutes, they make money from undergrad programs. Even the JIS University, they have their undergrad programs, they have their engineering colleges through which they make money. This is the institute out of the vision that I showed in the video you might have missed. It was very disturbing today. So here, the vision of our director was to make a postgraduate program. So that normally doesn't allow the funders, the funding group, JIS Group, to make money at all, because it doesn't come from master's program at all. It comes from the bachelor's program. So the idea is to develop a kind of top-notch institution in India, which is not present everywhere, especially not present in the Eastern Zone. There could be some IASCs and something, there are IASRs in different places, but not many at all. So that is the main goal. So we are looking for motivated students. So if you think you are motivated enough, you are meritorious enough, you want to really pursue your goal in the field of medical biotechnology and bioinformatics, you should come to us even if you don't have money, because there are scholarships available for the meritorious students. So it's, again, even if it's a private institution, normal feeling is that private institutions charge high amount of money. Definitely, it's a considerable amount of fees you have to pay. But if you are extremely talented, extremely meritorious, we offer scholarship to get high quality students and to build our institution together as a topmost one in India and at the world level, definitely in coming years. Okay, I'm fighting like anything. So now this was the thing, Microbial Evolution, Infectious Diseases and Computational Genomics. That's the title. So we now will go to each of these terms one by one. Say, first of all, microbial evolution. I don't know what to do. I'm just cannot control this thing at all. I wish I could have time to switch this thing to another machine, but that will spoil everything, I believe. Okay. So microbial evolution refers to the change in heritable traits, or if you can say evolution is the same thing. It refers to a change in heritable traits in a population. It could be microbial, it could be eukaryotic population, anything, over generation, caused by the genetic material, which mostly is DNA, but it could be RNA for some viruses, as we all know, say for SARS-CoV-2, causing coronavirus. Now, the thing is, normally people, when talk about evolution, there is a very kind of prominent misconception. Evolution is nothing but natural selection. That's what people think. It's not. Natural selection is only a byproduct of evolution. Evolution is continued. Natural selection happens because of some selection pressure and also accidentally. But evolution continues. Evolution is just a function of change over time. So these are some of the misconceptions. So evolution is not about things getting better and better and more complex. Selection is about survival and reproduction. Organisms with traits that help them survive long enough to reproduce will pass their genes on. So the next thing is, evolution is also not about organisms trying to adapt. Let me switch to a different mouse if it helps. Let me see. Just okay, so evolution is not about organisms trying to adapt. Okay. Trying does not change genes. Only heritable traits already in a population can be passed on. Okay. So only the inheritable traits that can be inherited, that traits will be passed on through generations. And also, evolution does not give organisms what they need. Evolution is a continuous process. Evolution doesn't know what organisms need, what they don't need. Okay. If this was true, that organisms, whatever organisms need is being supplied by evolution, then there would never be any death, any extinction of species. If individuals have traits that help them survive environmental change, they may pass those traits on while others die. So these are called the positively selected traits, adaptive traits. These adaptive traits, if they're there, so to help the individuals adapt, they will be passed on through the next generations which will survive, but the ones which do not have adaptive traits or which have maladaptive traits, I mean, there's some traits which are detrimental to their fitness, or when they are being outcompeted by the adaptive ones, they will die from the population. They will be wiped off from the population. Also, evolution does not mean that individuals in the population slowly change. Changes happen to populations over generations, as those with unfavorable traits are wiped off or weeded out, and those with favorable traits are more likely to reproduce. But this is when this kind of favorable and unfavorable traits we're talking about, then we talk about selection. But evolution is nothing about selection. Evolution is much bigger than selection. Evolution includes changes which could be favorable, which could be unfavorable, which could be doing nothing, basically. There is no such kind of fitness change with some kind of change that you see through evolution. We'll talk about that later. Evolution also not a theory of how life began. It's about how living organisms change over generations. So these are some of the misconceptions. So once we understand what evolution is, or what, say, microbial evolution, which is part of evolution, definitely is, then we come to the infectious diseases. But before infectious disease, we need to understand what disease is. Disease is not something that results from any physical injury, but disease actually is a disorder, some kind of discomfort of structure and function in an organism. Some kind of problem you're facing, producing, it's still kind of, it's making me crazy, producing specific symptoms and affecting specific locations. So that is what is called disease. Now, when the diseases are caused by microbes, which are pathogenic. So if a microbe is causing a disease, we call this microbe harmful to us or wherever it's causing the disease. For example, there are many microbes which do not cause any problem to us, but cause problem to some plants or to other animals. So they are pathogenic to those plants or those animals. And there are many organisms, many microbes that are pathogenic to humans. So we are more concerned about that. Definitely we are concerned about other plant and animal pathogens, as long as we use those plants and animals as food or in some other ways. Because people are all animals are very selfish. They think of themselves. So we are only concerned about those pathogens as long as they cause problem to us directly, that is when they are called human pathogens, or indirectly when they cause problems to plants or animals that we use as food or as, in other forms, actually. So these microbes, bacteria, viruses, fungi, protozoa, or helminths, or parasites, they could be all different forms. There are many examples. There's nothing necessary. It's not necessary to go about all these things. That's not the topic of today's presentation. And here is just a glimpse of the global example of emerging and re-emerging infectious diseases. Okay, you can see the blue ones. So first of all, the red ones, they are newly emerging. It's a 2017 picture. And blue ones are re-emerging or resurging. That means that they were there previously, they somehow got wiped off or somehow got subdued, but they are now again causing problem to us. And there is another group deliberately emerging, meaning that they are basically lab mediated, so human induced it deliberately. So that is one example. You can see here that's the anthrax bioterrorism. And people also initially thought that this SARS-CoV-2 could be this deliberately emerging thing, but there is no such evidence as of now. And I just do not want to go political at this point. So anthrax bioterrorism is there, but otherwise mostly newly emerging and re-emerging diseases you see, and it's kind of causing every now and then all over the world. And bacterial infections, I'm now shifting to bacteria out of all the microbes, because I'm just going to give you just a glimpse of this infectious diseases and how we can use computational genomics thing to understand this information. So you can see our body, and there could be eye infection, infection in nose, infection in inside the oral cavity, inside the respiratory tracts, inside our lungs, in the gut, you name it. I mean, intestine, vagina, urinary tract, you name it. So all kinds of infections could be there by different pathogens. And they are not always pathogens. For example, E. coli, they're mostly non-pathogenic, but sometimes there could be some kind of mutations which could cause problem to us, or when they move to a habitat where they are not allowed by the host species. For example, urine is normally sterile. You could have some kind of non-pathogenic bacteria here and there in urine, but normally it's sterile. So it could be such that accidentally E. coli population could move from the gut to this urinary tract and causing some kind of infection there. So that makes it pathogen in that particular habitat, right? So once we understand what infectious diseases is, this term is, then we move to the third phrase that I used in the title of today's talk, computational genomics. So this is, as you all understand, it's a subset of bioinformatics and computational biology, which actually focuses on genes and genomes to understand the principles of how the DNA of a species or RNA of a species controls its biology at the molecular level and beyond. Okay. So now, what is bioinformatics or computational biology? Well, you can see this biologist and computer scientists, they merge to develop this bioinformaticians or computational biologists. So again, I remind you that you can be only biologists, and you can use some of the, I'm sorry, it's really very disturbing for you as the viewers, and also disturbing to me not to be able to control this mouse. And it's something to blame my computer, I believe. Sorry about that. So as a biologist, you can use programs developed by computer scientists in a very meaningful way and become a bioinformatician. You don't need to learn computer programming. Definitely, if you learn, you can develop your own program, and it would be additionally helpful to you, but it is not mandatory. So don't get scared that you have to learn computer programming. Definitely, you can learn some basics, but you have to be a programmer, good quality programmer, to be a bioinformatics person. That's absolutely not true. Okay, now, like when we talk about this genomics thing, so it all starts and ends at nucleotides, the components of genes and genomes. We know there are 4 chemical compounds: 2 purines, 2 pyrimidines, adenine, guanine, cytosine, thymine. So when we do computational genomic study, this is how it looks like. You get this sequence of genes and genomes, and that is how you have them on a computer monitor, and you start using your analytical tool to analyze them and understand the biological meaning hidden in this entire code of nucleotides as part of genes or genomes in the organism of your interest. That is how you do it. Now the connection. Microbial evolution gives rise to changes in genes and genomes of microbial species. Very simple. And a fraction of these changes are linked to infectious diseases caused by the microbial pathogens. Again, please be assured that there's only a small fraction of changes. Okay. And most of the changes are not causing any kind of problems to us. Because if it would be the other way around, then we would have been extinct as a human race long before. And computational genomics is a branch of bioinformatics and computational biology that can be used to detect such link. That is a link between the evolution of microbes and the infectious diseases. That is what you can do here. And that's why if you learn this topic, you will have lucrative offers from, if you want to go to industry at all. Otherwise, you can be academicians like us. You can train the next generation. You can teach the next generations. And then that is how you can work, basically. That's not a problem at all. But if you want to go to industry, Then all pharmaceutical industries need this kind of expertise. You can all understand that. And okay, let me try. I'm just shifting to another keyboard. If the keyboard is causing a problem, I mean, that is how also the computational biologist would work, trying to use this analytical tool or that to find the link. If let me see if this helps. It seems a little better. Let's see. Okay, I believe we could solve the problem. Let's see. No, it's the same thing. I don't know what's going on. Okay, please bear with me for the rest of the talk if you want to continue. Otherwise, you can sit by to me. Well, so this is the link. I mean, computational genomics kind of connects the link between evolution and infectious diseases through the understanding of the hidden codes in the DNA or RNA as genetic material. Okay. So that's all of my talk. That is the gist of the talk. Now I'll go to each and every detail. Again, coming back to the title. So the talk really begins here. So whoever didn't join earlier didn't miss much. All was introductory stuff. Now, as we talk about microbial evolution, we should start from understanding the age and magnitude of microbial diversity is huge compared to us and humans a few thousand years old. Here they are kind of 3.5 or more billion years old in age. Okay, and if you have these numbers, you can see number of stars in the universe is 7 times 10 to the power 21, and number of microbes on Earth is 5 times 10 to the power 30. You can understand the difference. I mean, just on Earth, how much was the amount of microbes that we have. That shows the diversity of microbes as species. We will next talk about diversity of microbes within a species, how much that also could be high. Okay, so, oh no, it's just not helping me at all. Okay, so there are many other values. I just don't want to go into all this information because in the last couple of webinars, my colleagues have talked about the numbers of microbes. It's way more, 3 times more than the human cells are there in a human body. So it's, I mean, it's huge. You can understand that it's more a problem, if we don't have these microbes in our body, than to have an organ, because it's like microbiome is now considered as a specific organ for specific part. I mean, there is oral microbiome, that is organ in the oral cavity, gut microbiome, that is an organ in the gut, skin microbiome, there is an organ in the skin. So you have to have, or we have to have, a balance in the diversity and quantity of these microbial species there, the non-pathogenic or the commensal microflora, that the balance need to be there. Otherwise, it's a big problem, huge problem if there is any disruption going on. That is how this probiotic thing has come up in terms of medicine. You get, so you have some kind of disruption problems in the gut. You have this bacteria, millions of bacteria. You just swallow the millions of bacteria so that these helpful or the commensal microflora would try to outcompete the problematic microflora that are there at present in your gut. Okay, so coming back to evolution, that is the normal thing. I'm not talking about microbial evolution at all here in this slide. is a function of variation over time. And when we see this function of variation at the molecular level, that is basically the alteration of existing sequences, whatever sequences you're having, if it's changed, that is the variation, right? And that actually leads to the variation in the genome sequence. Now, there is something more in the microbes, a little more, and that causes a huge difference. What is that? That is, in addition to alteration of existing sequences, there is another thing, inclusion of new sequences that you don't see normally in eukaryotes. You can see in some cases, in some protists, because of the fact that they phagocytized, when they phagocytize some kind of bacterial cell, they lyse them, and then the bacterial DNA somehow accidentally get incorporated in their genome, and that could be called inclusion of new sequences in those protists. That is the kind of major model of this kind of acquisition of new sequences in eukaryotic genome. Otherwise, in eukaryotes, you don't see this inclusion of new sequences thing. And that causes a huge difference so far as the microbial diversity is considered. Now, let's forget about all this huge diversity we're talking about, these numbers, different bacteria within a human, different bacterial species within a human body, all these things. Just consider only a single species. And one of the commonest one, we all know this name, Escherichia coli, commonly we call E. coli. And we're taking here the genome, say each circle here, you see a Venn diagram, where the circle overlaps with each other to some extent, and there are some unique position. There is something that I do not want to show right now, and it's coming up automatically. Okay, sorry. So here are the three genomes we're considering. That is, one is MG1655 or K12. It's a non-pathogenic strain. It was first isolated from a student of class 12 in the US. That is how it was termed K12. EDL933, this is also a gut E. coli coming out of feces, but it is causing bloody diarrhea. It's a very problematic enterohemorrhagic E. coli, it's called. And the third one is CFT073, uropathogenic, causing cystitis, that is causing infection in urinary bladder, and that is what is called cystitis, and the CFT. So you can see they have genomes of the same species. It's one single species, and two of them are from gut. And you can see the amount of common genes. See, if each circle represents the number of genes, the gene content in their genome, then you can see only 40% of genes are in common. And these are the other fractions, which you see that, for example, 7% you can see, which is common to both MG1655 and EDL933, and then 2% common between K12 and CFT, and so on. So you can see the huge diversity of gene content, how this happens. We will talk about that briefly. It is what is called horizontal gene transfer. We'll talk about that in a few minutes. But before that, this will give you a glimpse of diversity of E. coli or a diversity of bacteria within a single species. And so you can tackle both. When you study computational genomics, you can study pangenome. Pangenome means when you take all genes in all genomes of a bacterial species, given whatever sequences you have in hand. Like at the population level is good, but if you don't have whatever number, 3, 4, 5, in 3, 4, 5 genomes, whatever number of sequences, genome sequences you have, among them, if the genes are present in all, say, 5 genomes you are considering, they will be called core genes. If the genes, for example, say you are taking only 5 genes, or maybe just the 3 genomes, you're taking only 3 genomes. if the genes are present, if there are genes which are present in all three genomes, they will be called core genes. That is, genes shared by all genomes. If the genes are present in only two of the three, that will be called genes shared by some genomes, the mosaic. And the genes that are present in only one of them, that is called the genome specific or strain specific or unique genes. So these genome specific and mosaic, these together are termed accessory genome, and core is there. Together, core and accessory form the pangenome. That is what you saw in the slide before when we were taking three different E. coli genomes. So there are sequences which are specific to, or genes specific to genome A or genome B or genome C. There are genes which are specific to only two of the three, and there are genes at the center which are present in all the three genomes. So now you can increase the number of genomes and you can calculate the pangenomic profile of a bacterial species. So that is very important to understand the information, or understand the movement of genes from one species to another, how genes move from one species to another or from one strain to another strain within a single species. So, and what are the genes that are there which are probably doing some such important stuff that they need to be present everywhere. So they are core. That is also a very important thing to know for us, the core fraction. That is, normally, for example, we call some genes housekeeping genes. They work, they do some kind of basic metabolic and physiologic functions in a cell, so they need to be present in every species, every strain of a species. So they are normally core genes. So together, you can build a pangenome. Now let's consider horizontal gene transfer. In some cases, actually, I'm moving to the next slide just because automatically the slide is being moved by some ghost or some kind of magical thing is happening here. I have no control, so I'm moving my discussion also to the next one. I just wanted to talk a little bit more on the pangenome and then move to this slide, gene transfer. So gene transfer can be vertical and can be horizontal. Vertical thing is what you see from one generation to the next generation. That is sharing the common ancestry. But what I'm talking about, this mosaic or genome-specific genes or accessory fraction, that is what you get through horizontal gene transfer, not through vertical ones. Horizontal gene transfer, strict definition is acquisition of genes from a different species. A loose definition is acquisition of genes from a different organism. But normally, when it's from different species, we call them horizontal gene transfer. Now, here I'm showing you species 1, species 2, and maybe some other species here sitting. And that is how the gene moves from one species to another. That is what is called HGT in short, that is horizontal gene transfer. However, so these are, for example, these are the two strains of same species, two isolates of same species. These are like 1, 2, 3, 4 isolates of the same species. So now here is B, here is B, here is B. It's the same gene. They're called orthologous genes. So now it could be such that a fraction of this moves from this particular isolate to this particular isolate. That will be called horizontal gene transfer. That will be called recombination. That is known as recombination when it is happening between homologous sequences. That is what is known as recombination within a single species. Homologous sequences within a single species, that is what is known as, you can see recombination happens within species, and that is kind of one fraction. Say, I'm showing you one example here. Say, these are the two sequences that recombine to one another. So they're called, one is called minor parent, one is called major parent, based on what fraction of these regions are coming to the recombinant or the daughter cell. So this is the recombinant DNA. And you can see this, only this fraction moved from here to form a recombinant DNA. So that's why, say, if these red lines are denoted by mutations, you can see these mutations, you can find the mutations here too. Okay. So this is the recombinant DNA. Therefore, you can see this recombinant, the action of recombination happens within the homologous genes of a single species, whereas if it moves from one species to another, and a large, horizontal gene transfer actions as a kind of large fraction of genes, many genes together, few kilobases of regions moves from one species isolate to another species isolate. That is how it kind of generates huge amount of diversity, which cannot be possible only through mutations, which normally you see in eukaryotes, and that's why the eukaryotic diversity is kind of no way comparable to the diversity you see in the microbes. It's because of this horizontal gene transfer. Now let's move to the other thing which is applicable to all kinds of evolution, microbial as well as eukaryotic, that is this alteration of existing sequences. So sequences are already there. There is no acquisition of new sequences, but there is mutation or some kind of changes, or maybe deleted. Deletion is also kind of mutations. Some regions deleted, and that actually lead to some variations in the genomes and gene sequences. And that is another type of microbial evolution. So here, again, the same problem, cannot control. Microbial evolution is a function of variation over time. We already saw this. And at molecular level, it alters existing sequences, meaning that it mutates DNA, RNA, or protein sequences, leading to different alleles. So first it kind of changes, it brings changes in the DNA or RNA sequences, and automatically it changes can be, not always, I will talk about that when these changes can be reflected to protein sequences and when it won't be. But it leads to, whenever you talk about these changes in DNA, RNA, or protein, it would lead to different alleles of same genetic region. Say, if we consider a gene, so different alleles of a gene sequence. So for example, if you have a gene sequence from 10 individuals, so you get 10 gene sequences. So 10 gene sequences doesn't mean that you have 10 different alleles. All the sequences from 10 individuals could be identical. So you are coming up with a single allele, or they all could be different from one another. Then you can get as many as 10 different alleles. So the number of alleles can vary from 1 to the number of sequences you are considering. Now, selection or not, you have to, if one mutation, so for example, so there is a mutation in DNA, RNA, protein sequence. So one mutation means one new allele. And then one new allele compared to the existing allele. So this is the mutation that brings in the new allele in the system where there are already many other existing alleles. So now the question comes of fitness. Whether this new allele is more fit or less fit, or has no difference compared to the existing alleles. And that is where comes the term of selection. More fit means positively selected, no difference meaning neutral selection, and less fit means purifying selection. Okay, purifying means, why it's purifying is it can also be negative selection. It is called negative selection. It's purifying because if it's less fit, then kind of through natural selection pressures, it will be, such a variation or such a variant having this less fit variation will be will be perished or will be wiped off from the population, thereby purifying the population. So population will try to have the most fit ones, or at least reasonably fit ones. The population of any organism doesn't want to have an unfit one. That's why when as a kid, our parents always talk about, like, you have to learn this, you have to learn that to be more fit to the community. In the population, in the human population. So we are teaching, we are kind of, as a student, we are learning something just to become more fit. So everyone tries to be more fit. If it's less fit, then you are basically getting out-competed by the more fit ones, and then nature automatically wipes them off, and that is where population is automatically or naturally become purified. Therefore, this kind of selection is also known as purifying selection. Now, let's think about this selection, how this positive selection works in terms of virulence evolution. Say, let's consider these green lines are kind of an epithelial tissue in a particular habitat, where there are these receptors, this circle, this line, and these circles. I hope you can see my cursor here. They are receptors, and this is a bacteria, and this red structure, semicircular structure that you see here, that actually they're called adhesin molecule, which help the bacteria adhere or to connect, to bind to the host receptor molecule. Okay. So let's, for example, take a real scenario. See, E. coli. E. coli is a commensal one in the gut. It's a commensal habitat, is the gut in human host. So let's consider this green region is a epithelial tissue in the gut, and the E. coli, one E. coli population is bound very nicely, they're bound to the receptor. Now, accidentally, again, accidentally, E. coli, no one goes to a situation which is completely new, where you have to struggle and all these things. No living organism wants to do that. E. coli also doesn't want to do that. But accidentally, if it moves to a different habitat, say urinary tract or urinary bladder, somehow it can go through your feces, or when you clean your feces automatically, somehow, if you are not cleaning it properly, especially it happens for the girls, it can enter the urinary tract. Now, in urinary tract, the epithelial tissue has a different kind of receptor, where it cannot bind as nicely as it was binding in the gut. So it's now low fit. So now it starts mutating even more. It is mutating, but there is a selection pressure acting, but mutating randomly. This E. coli doesn't know which change specifically will help it to bind stronger. So it will try generations after generations, all possible things. So that is how, say it could be such that there is a acquisition of new gene. Say here, there's a new gene coming here, and this new gene is having is coding for a protein that kind of developing this new structure of new adenosine molecule, which can bind strongly enough. So that's a different adenosine molecule present in this genetic material, which I show here as plasmids, for example. You know what plasmid is. It's a self-replicating molecule. You can see, apart from the bacterial DNA, separate from bacterial DNA. So it is there, and it can take the charge of this original adhesin molecule. And it can take the charge even more when you can stop the expression of this gene, so that you will see these proteins here, these old adenosine molecules here, because they are taking the space. So you can have the gene loss. Gene loss means it might not be lost from the genome, but it has formed, it is called pseudogenization. So there is a mutation that disrupts the expression of this gene. So gene is no more expressing. So you don't see this kind of structures that you used to see here in this new bacterial cell. Rather, this is taken up by the adenosine protein molecule coded by this new adenosine gene. Now, this is I'm showing only one change, but there could be, to adapt, you need to change many other genes in the genome, mutate in different ways to make yourself more adaptive. And another way is that instead of having a new gene, instead of acquiring a new gene, you can have a mutation. You can have a mutation in the existing adenosine molecule that can change the semicircular protein structure conformation to this triangular shape, which can now bind very nicely to this new receptor molecule in the urinary tract. So that is another way of adaptation, which can lead to virulence in urinary tract, because in urinary tract they are actually it's actually sterile, so they are not allowed to be there. So they are not use urine as a common habitat. So it causes problem to the host, to the human, basically. Now there are a lot of changes are happening here and there to make this bacterial population, which have accidentally entered this urinary tract or bladder, to adapt to this new system. Unfortunately enough, evolution or selection doesn't actually look long term. They have no foresight from that perspective. It is selection happens to help the organism get any kind of immediate advantage. So when they're having this immediate advantage, now when they're coming out of this urinary tract, in whichever way, say for example, they get flushed out, or they antibiotics are used, so some whichever way, these subpopulation of this bacteria coming out of the urinary tract, and then they are unfit anywhere else. So if they want to go to their normal habitat in the gut, so they will be not fit enough. They will be outcompeted by the existing population, this population there. So this is causing an immediate advantage, but there's a functional trade-off effects which will not allow it or this population to survive well enough in the outside environment. That is a very common evolutionary dynamics. We are not going to the dynamics. It's called source sink dynamics in microbial evolution. So, but what I'm trying to say that genomic information, genomic information is this way used to understand the evolutionary mechanisms, dynamics of a population. Here we're talking about microbial population. Now, how to detect selection? And obviously, it's all about the diversity, the changes. You have to map the changes. You have to calculate in whichever way the changes that are happening in an organism. So definitely, there are two kinds of, broadly, there are two kinds of genetic regions in a genome. One is the protein coding genes, which code for proteins, and the remainder, the other regions. Okay. So we are not, there are tools to analyze both kinds of DNA regions, protein coding and non-coding, or coding for RNA genes, say transfer RNA or ribosomal RNA, because it's available for different regions of DNA, but I personally want to consider here just one tool, and the most powerful yet very conservative tool today, because that is what has changed the world of detecting selection in molecular evolution. So I want to discuss that. And another reason is that in protein coding genes, you have an additional information. to analyze. In the next slide, actually, we'll talk about this. What is this additional information? Nucleotide sequence bear changes, but when the nucleotide sequences code for proteins, then you have this additional information of triplet sequences that we call as codons that actually translates to a particular amino acid. That is the additional information you get for protein coding genes that you don't get in non-protein coding genes. And these two types of, so there are two types of variations or changes that you see. There are changes which do not alter the corresponding amino acid, and there are changes which changes the corresponding amino acid. Why it is so? Because, so there are triplets and there are 4 nucleotides, A, T, C, G. So 4 to the power 3 combinations, so 64 combinations are there. So 61 can code for different amino acids. There are 3 codons code for stop codons, but amino acids we have are only 20. So there is something active in nature what is known as degeneracy of codon degeneracy, which means that there are multiple codons coding for the same amino acid. And that is where you get, that is how actually you get 2 types of mutations in protein coding genes. If the codon changes, if one codon changes in a way that it forms, it's another degenerate form. So for example, here, GGA stands for glycine and GGG also stands for glycine. So when there is a mutation in the 3rd position of this GGG, which changes this triple G to GGA, then basically it is forming, it is changing to its another degenerate form. So the corresponding amino acid is not being changed. However, if the nucleotide changes or the codon position changes in a way that's from GCA to GTA, which changes the amino acid from alanine to valine, its non-degenerate form, then we see a different kind of mutation. So you understand from the name that the amino acid remains the same in this kind of changes, so that's why they call synonymous change, or silent, that is structurally silent. There is no change in the structure, protein structure, right? Everything we think from the perspective of protein, because functional molecule is the protein, right? So we are only interested about the ultimate fitness of an organism, how the protein functions there and how the protein function can change the fitness of an organism. So from that perspective, the synonymous changes are, to you, it can be something like that you consider that synonymous changes are not that important. But in the next few slides, you will find that synonymous changes actually are even more important than the non-synonymous ones, which actually cause replacement of amino acids. But at this point, definitely, from this slide, you can understand that non-synonymous ones are extremely important. And here is the codon table, synonymous and non-synonymous sites. You can calculate 61 codons, I told you just now, for 20 amino acids. Second position, any change in the second position always leads to non-synonymous change. There are degeneracy at different levels. I mean, there are amino acids which can code for there are codons which can code for, 6 codons code for single amino acid, that is called a 6th-level degeneracy, or there could be 4 codons coding for single amino acid that we see for alanine, glycine, proline, threonine, valine. There are 3 codons which code for single amino acid, that is isoleucine, and there are many codons, many cases where single amino acid is coded by 2 codons. So there are levels of degeneracy like 2-level, 3-level, 4-level, and 6-level degeneracy. The only two non-degenerate codon we see is methionine and tryptophan. These are the two non-degenerate codon because in such case, there is only one codon that is coding for one amino acid. There is no other second codon coding for the same amino acid. Now, if we calculate synonymous and non-synonymous sites, you can calculate in a way, I'm going pretty fast here because just to give you a glimpse, you might not understand it completely, and especially in this case when mouse is being uncontrolled, I cannot control. I mean, you see, for example, it's just moving automatically. So how it's calculated? You can see the variations, you can observe the variations, say if there is a change in the sequences. But you can also find out what could be the estimated amount of variations. So for example, if there is say for example, you take the GGG. There is a codon GGG coding for glycine. So at a time, if you consider one single mutation, so you can find there are three different possibilities at the third position or second position or the first position. At each position, there are three more possibilities of mutations, right? From G to A or C or T. So now you can calculate if, for example, from this codon table, there is a mutation at only the third position, say from GGG to either GGC or GGT or GGA. In all cases, it's synonymous. So the third position, any change accumulates in the third position is going to be synonymous. So that is how you consider this third position as a synonymous position or synonymous site. Second position is always non-synonymous, I told you already. Any change, you can just look at the codon table. If you can find any synonymous change, let me know. It's always non-synonymous. In the first position, if you change this G to A or C or T, it's always non-synonymous. So you find that the first and the second position, these two positions are non-synonymous, and the third position is synonymous. So that is how we can calculate in this codon, there are two non-synonymous sites and one synonymous site. For example, you can see the number of synonymous sites, 1 for this codon, and number of non-synonymous sites, 2 for this codon. That is how you can calculate for each sequence total number of synonymous sites and total number of non-synonymous sites. Why we want to do it, I'm going to explain in a minute. So similarly, you can do it for the second sequence, and you can get these values, for example. Now, if you compare the two sequences, now you want to see the variation in the two sequences, and then you can have the average synonymous sites, just 3.33 plus 3 divided by 2, that is the average synonymous sites for these two sequences, and equally you can calculate average non-synonymous sites for these two sequences. So these shows that these are the number of positions, average number of positions where either synonymous change or non-synonymous change can happen. Can happen. It's not that it is happening. It can happen. Now, what are the changes that we see here? We have these two sequences, and we see that number of synonymous mutations, 1 here, the first position is non-synonymous here. The second codon, there is one non-synonymous mutation, and third codon, you see one synonymous mutation. So we see the observed number is 1 for synonymous, 2 for non-synonymous. Now, we can calculate the proportion of synonymous differences and the proportion of non-synonymous differences by taking the ratio of the observed number of mutations to the number of mutations that can happen. That shows the rate of changes, proportion of changes. So rate actually, initially it used to be calculated like this. This is the way it is calculated, but Jukes and Cantor, two scientists, they came up with a correction that is called the Jukes-Cantor correction, That is the final rate calculation, which is called the rate of synonymous mutations and rate of non-synonymous mutations, DS and DN. Why do we want to calculate this? Again, we want to know whether the non-synonymous mutations are occurring at a significantly higher rate. But how do you know? How do you know? I mean, because we are interested in non-synonymous because that changes the protein structure and possibly protein function. So that's why we're interested in these non-synonymous changes. Quite understandable. But how do you calculate it? There, actually, we take this synonymous rate as reference. That's how synonymous changes are becoming extremely important. Okay. So the rate of synonymous mutations being, synonymous mutations being kind of silent mutations, protein function and structure wise, there is no direct influence of synonymous mutations in protein structure and function, so they can be considered neutral, and they are found to accumulate at a constant rate for a given gene. So if you can use this rate as reference, then if the rate of non-synonymous mutations you can find is significantly higher than this reference rate, then you can definitely say that particular sequence is undergoing a strong selective pressure, undergoing some kind of positive selection for the accumulation of amino acid changes. However, one caution here, the caution comes here. The caution is the synonymous mutations can be adaptive too. Okay, but this is definitely a rare scenario. In most cases, it's still accepted view that synonymous mutations are neutral, but definitely you have to be cautious about their possible adaptive nature. So synonymous mutations being neutral, you can see these spaces. I'm not going into much detail here, but you can see these DS values. It actually shows the timeline, comparative relative timeline of their emergence. So DS estimate actually serves as a molecular clock of the emergence of different species. For example, E. coli and Salmonella, they emerged or they diverged from other enterobacteria at around the same time, around say 30-40 million years ago, something like that. But the most important thing here, we have only subspecies 1. So that's why you don't have the DS value comparable with E. coli. It's much lower. And it's only a single subspecies of Salmonella which diverged much later than the entire Salmonella species. So Mycobacterium tuberculosis is very recently emerged, and even more recently emerged the Yersinia pestis. You can see from these values their relative ages, and it's in thousands and millions of years. When we talk about evolutionary time, it's not about days or months. In contrast, non-synonymous mutations, they're relatively few are neutral, and most of them are detrimental. And very rarely they can be adaptive. Very rarely they can be adaptive. Okay. So mostly they are detrimental. And you can see there is not much information here going on in the DN value as in terms of molecular clock. So DN over DS is used as a very powerful statistic to identify selection. It was first shown by two Japanese scientists, Nei and Gojobo, in 1986, where DN over DS, this ratio is significantly lower than 1, meaning that there is no pressure, no selection pressure acting for the accumulation of amino acid changes. So that is where you talk about purifying selection or negative selection, where it is more or less equal to 1. I mean, there is not much difference between DS and DN. It's noted frequently in pseudogenes, meaning that when actually there's a loss of function in the genes and there is no selection pressure acting, no kind of selection pressure acting on the gene. So mutations are happening every now and then at the same rate as that of synonymous mutations, and that's where the rate becomes more or less the same. That is what is called neutral selection. And the very rare situation that you see is that the DM is significantly higher than DS, suggesting a selection pressure, especially for the accumulation of amino acid changes. That is where we talk about positive selection or natural selection. It is only a small part of evolution. It's powerful but very conservative, and you see the fraction of genes with DM over D is significantly higher than 1. You can see only E. coli, 2.3%. Yersinia, it's kind of almost 1/6 of genes, but this could be from completely from different perspective. Normally, the newly emerging species showing this kind of DNDS greater than 1 doesn't directly translate to the positive selection. There could be another kind of selection, it's called relaxed purifying selection, but that's a different thing. I'm not going to that detail, but what I'm trying to say that E. coli being a kind of great grandfather of all these microbes, you can see a salmonella, you can see even lower fraction of genes, DNDS greater than 1 is just 1 to 2%. Very few genes under positive selection. And that is because of these problems. The thing is that there could be selection at some point, at some region. So when you talk about a gene sequence which codes a protein, not the entire protein is under selection. Only a few specific regions are under selection. Okay, always keep this in mind. Not the entire protein, and entire protein cannot be in selection. Only a few residues which are critical from the perspective of structure and function of the particular protein could be under selection. So, for example, those regions, we are talking about, say, genes in those regions of proteins, genes coding for those regions of proteins might be under selection long ago. But afterwards, there could be many other neutral mutations happening here and there across the gene. So now this is the data set we are having in hand. We're not having this older data set. We're having these are the data set. This is kind of evolutionary past information. We don't have them in hand. We have this information. So here, these red ones, there's a selection indicator of selections, will be overshadowed by these blue ones when we calculate the overall DNDS value for the overall gene or overall protein. So the same scenario can happen when there is a recent selection. It could be such that there is already kind of an array of neutral variations accumulated across the gene. Now it's kind of disturbing throughout. A lot of neutral variations accumulated long ago. Very recently, some regions, some residues could be under selection, and they accumulate some variations which are positively selected, but they will be totally ignored when we calculate the entire gene, DNDS for the entire gene. So that's why there are some modified versions of this DNDS calculation. We can consider specific gene regions. We can use a sliding window that we are calculating DNDS for a particular size of window, say 100 bases at a time, and then moving 50 bases, and then say from 1 to 100, and then 51 to 150, then 101 to 200. That is how we're moving, and then we are having the values of DN and DS. There you can find that there are regions which are showing much higher value of DNDS compared to DS, and there are many other regions which are not showing this kind of signature. And you can calculate DNDS for individual codons or you can have this information. If you know well about the genes you are studying, you know that these are the genes which, I mean, this is a gene from the organisms which are not supposed to be under selection, but we have these organisms, the same gene from these organisms could be under selection. Then we can use some kind of what you call background branch, foreground branch, and background branch is under no selection. We can have this null hypothesis, and foreground branch is having under selection. So that is how you can calculate it. You can find out whether the null hypothesis is working or not. So again, there are softwares for that. We don't need to do that. We just need to understand the biology. You can create your own software tools. Don't get kind of terrified by this all kind of statistical information that this null hypothesis and all these things you have to do. You have to perform maximum likelihood calculation and all these things. Those all can be done by the software tools, but you need to know the biology and you need to know how the software works. That is all you need to know, but you don't need to know any programming language to run those, to create those softwares, because you can run the software from scratch. However, even then, there are many drawbacks of DNDs. More or less, it's conservative. For example, this one, this lineage-specific DNDs or branch-specific DNDs is highly powerful, but you hardly can do this for the entire genome. And especially when you have, say, hundreds of genomes, it's just impossible to do because it's very rigorous, statistically rigorous, so it takes enormous amount of, it's a very computationally intensive process. You cannot do it easily. It will take months to complete your analysis. So there are different alternative ways to look for selection. Today I will just discuss one other way, and that actually again arising from what we see normally. Convergence, morphological convergence. So for example, well before humans migrated from one place to another, when we used to feel cold, we somehow protected ourselves using some form of warm clothes, whichever way we made them. So that is kind of to adapt to a similar environment. Say a particular weather, we have to adapt to a particular weather. Accordingly, we change ourselves. Especially here, you see that is kind of one normal scenario. You see that convergence in human nature, you see convergence in human society, you see that at the molecular level or at the biological level, if you look for convergence. So this is the macroevolutionary level you can see here, where you can see this evolutionary pathways. Fish emerged well before these mammals, and then you can find that there is a mammal coming up with the structures which are similar to the structures you found in fish. That is because that this mammal, for some reason, wanted to or had to adapt to the similar environment, that is aquatic habitat. Fins or flippers, they do the same thing. Like even for ducks, you have this webbed feet-like structure to help them swim. So that is what is called convergence. So researchers try to find out whether, we can use this convergence at the micro level, at the molecular level. Now, what we found that this, if two organisms are trying to adapt in a similar environment to acquire a fitness, a particular fitness, then there could be some genes or some proteins which could be necessary to get adapted or to help the organism adapt to that environment. So the changes also could be similar there. For example, what I'm trying to show here, the adaptive mutations tend to repeat. Here you can see a particular protein molecule. It actually accumulated, say, a change at position 27, amino acid position 27 from alanine to valine. Here, there is another allele leading to a synonymous or silent change. But maybe somewhere else, I mean, in a completely different allelic background, in somewhere else in evolutionary time, some other evolutionary time point, if this mutation can offer any kind of adaptive advantage, that mutation will tend to repeat if this protein is also trying to help the organism adapt in the environment which is similar to this. So if we take this from the other's perspective, that if we see that we are taking gene sequences, I mean a particular gene, a particular protein from individuals completely separate, geographically separate. For example, we are isolating E. coli from an Indian girl and a girl from the urine of an Indian girl and from the urine of an African girl or a European girl. And then we can, it is very often we have seen that same changes occur in some of the genes. For example, we talked about one adhesine gene, right? If you remember, adhesine genes are the ones which code for adhesine protein, which help the bacteria bind to the host receptor molecule. So there are specific host receptor molecules in the urinary tract. So when the bacterial population move to this urinary tract, they have to adapt. They need to adapt to this new environment. So there need to be some changes, some variations, some mutations in the adhesine molecule. The thing is, the changes in most cases, whether it's happening in the isolate from an Indian girl's urine or European boy's urine or somewhere else, you will see the same change tend to occur, because it suggests that this change, for example, in that adhesine molecule, will offer some kind of fitness advantage to that E. coli isolate or E. coli population to sustain in that urinary tract or bladder better. So that's why those mutations would tend to repeat. So why don't we find this convergence? That is kind of one very kind of booming area to find out or to track the footprints of positive selection through evolutionary convergence of mutations. For example, say this is a phylogenetic tree where, I mean, each node, say for example, stands for one particular allelic variant, and say along this branch there is a mutation from alanine to threonine at position 27. Here also you see another mutation, alanine to valine at position 27. Here also you could see another mutation, again alanine to valine at position 27. So it shows that they are unlinked mutations. They are not kind of coming from a shared ancestry. So it's the repeated phylogenetically unlinked mutation in the same amino acid positions. That is what we call convergent mutations. So we can study this convergent mutation to identify kind of very important insights, to have very important insights on the adaptive evolution in microbes and also in any organism. So here I'm showing you just one last example where we ourselves could use this kind of idea of convergent mutations to find very useful information regarding the fever typhoid in human. It's caused primarily by this Salmonella typhi and Paratyphi A or Paratyphi B. These are the ones that are restricted to human, causing this typhoid kind of disease. I'm not going to all this detailed information. There are two types of salmonella which can cause invasive diseases like typhoid kind of diseases, and there are servers which cause kind of normal gastrointestinal issues which can be self-limiting and self-clearing thing. But these are the ones like systemically invasive infections can be very serious, as you see for cases of typhoids. Now, what we found when we kind of developed a matrix to find out the number of genes, number of common genes that are having convergent mutations between these two, between any two salmonella servers. So here we took this only the invasive ones, say Dublin versus cholera Swiss, two groups of servers. Servers are the ones, showing a specific, one server showing one specific serological properties. So that is how salmonella species can be subdivided into multiple servers. So these are all different servers, and each server can, they are composed of many strains. So here, if you compare the this pair, Dublin and cholera Swiss, you will see only three common genes which are having convergent mutations. And this is how the value moves across any pair. When we look at paratyphy E and typhy, there are 37 genes, statistically way higher than any other pair. And remember that typhy and paratyphy A, already previous researches showed that they can actually, they infect the same host tissue in human, and sometimes they can co-infect. So it is quite understandable that they will use similar tricks, similar strategies to adapt to the same host tissue they are going to infect. So, and that is what probably leading to this high high overlap of common genes showing convergent mutations. And here I'm showing, like, then from the list of genes that are showing here, the 37 genes, I'm showing just only a few genes. You can find out the functions they do. And from that, you can get some kind of information, whether they are useful at all. For example, if you talk about this propionate, propionate metabolism, and this propionate metabolism, actually, they can actually, what happens in particular, this phosphotrans acetylase, or PTA genes you can see here, that represents this particular class of propionate metabolism, plays an important role in salmonella virulence, and actually this catabolism is necessary for the persistence of infection in human host. That is how this group, this functional group, is extremely important here, because repression of these, for example, repression of this PTA enzyme actually results in the accumulation of another compound called acetyl coenzyme A, or acetyl CoA in short. This allows growth retardation along with repression of signaling through the there is called a kind of two-component signal system. I believe a couple of days ago you heard about two-component system, 4P4Q two-component system. So repression of this gene is not something that is needed by the organism. Okay, so there could be mutation which can avoid such kind of repression. So similarly, signal proteins are there. These are all, known virulence-related functions. And for example, this ferrioxamine binding and uptake in association with the TOMB thing, they play a major role in intestinal colonization of the pathogenic bacteria. And similarly, we see many other genes which are well known to encode proteins that are important for survival and virulence of salmonella within the host. So that is how I mean, from the information of this convergent evolution, convergent mutation, you can come up with the understanding of the virulence dynamics of a particular disease, which we are talking about today. So now I will end this talk with a video which will show this is going to be about one lady who was maltreated, but who was also considered, or who still is considered the most dangerous woman in the US history. So we'll talk about that, how, and we'll know about this lady, and we'll know from this video how actually this lady is connected to infectious disease. And this lady is called Typhoid Mary. Let's hear this video.
In the early 1900s, germ theory was a relatively new area of study, and doctors were only beginning to understand how bacterial diseases like typhoid were spread. Poor Irish immigrant who worked as a cook for wealthy New York families was acting as a one-person super spreader, leaving a trail of sickness and death in her wake, which would forever earn her the name Typhoid Mary. Today, we're going to take a look at the true story of the woman called Typhoid Mary. When Mary Mallon was first quarantined in 1907, doctors determined she had already infected 22 people and caused the death of a young girl. By the time she was permanently quarantined in 1915, she had infected 51 people, at least 3 of whom had died. No asymptomatic carriers of typhoid had ever been identified at that time, so Mallon, who was always healthy, never believed she was the source of the outbreak. Nonetheless, public health officials repeatedly traced the frequent typhoid outbreaks right back to Mary, and despite a lifetime of resistance, Mallon eventually wound up in permanent quarantine. The first vaccine for typhoid was created in 1896. While it was a great help to soldiers who were at high risk of contracting the illness, it never became widespread enough to be of much value to the general public. So in the early 1900s, the disease was still extremely dangerous and had a mortality rate of about 10%. The symptoms of typhoid are not pleasant. 1 to 3 weeks after infection, a person suffering from typhoid typically contracted a dangerously high fever. This was quickly followed by nausea, vomiting, muscle pain, and headaches. Following that, a distinctive rash would appear on the victim's chest. If no treatment was administered by this point, the next phase would usually be intestinal bleeding, leading to blood clots under the skin. In some of the most dangerous cases, the abdomen would then be distended. In 1906, a homeowner whose family had endured a particularly violent and seemingly inexplicable outbreak of typhoid hired a New York State Department of Health sanitary engineer named George Soper to figure out what happened. Soper knew the incubation period of the Salmonella bacteria that causes typhoid was exactly 3 weeks. Starting his investigation by examining the cervix, his suspicions quickly fell on an Irish immigrant who had been hired as a cook precisely 3 weeks before everyone got sick. That person was Mary Mallon. Born in 1868 in what is today Northern Ireland, Mary came to the United States in the year of either 1883 or 1884. She started off living with an aunt and uncle and found work as a cook for well-to-do families. Soper ran a background check on Mallon and quickly saw that typhoid victims seemed to appear wherever she went. He also noticed she had a habit of changing jobs and names after each outbreak. Putting 2 and 2 together, Soper figured he had his woman. Soper questioned Mary and raised the possibility that she could be the carrier that was causing the typhoid outbreaks. Despite the evidence, Mallon denied Soper's allegations and resisted testing. No matter how many outbreaks followed her career, Mallon steadfastly refused to admit that she was the carrier of typhoid. Instead, she would quietly slip away after each outbreak, change her name slightly, and find a new job with a new family. Whether Mary actually understood she was a carrier of the disease and what is to be gleaned from her behavior is unclear. Her habit of changing her name could imply she was trying to deceive people into hiring her because she knew she was sick, or it could simply be a person's innocent attempt to avoid press attention. Typhoid at the time was typically associated with poor hygiene and, by extension, poor people. As such, New York's health officials were surprised to encounter the illness in wealthy and upper-middle-class households. Servants were put under the microscope, so to speak, and eventually they got around to Mary. Her poor hygiene did turn out to be an issue. When first questioned by health officials in 1907, Malin told them she didn't see any point in washing her hands before she handled food. This might sound reckless to us today, but in 1907, the theory of germs was relatively new, and Mary didn't seem to believe sickness could be transferred by physical contact. That being the case, she wouldn't see why hand washing was important. Scary stuff to think about. On one occasion in 1907, Dr. Soper came to Malin's workplace to take samples. Mary took this intrusion about as well as you'd expect, which is to say she completely refused to cooperate. In fact, one version of the story holds that Malin snatched up a meat cleaver and chased Dr. Soper out of the house. Other versions say it was a rolling pin or a meat fork, but no matter what kitchen utensil it was, the gist is always the same. It took several more tries before authorities were able to get the goods on Malin, and she never became more cooperative. Soper eventually planned on having her forcibly restrained so samples could be taken, but before he was able to carry out his plan, the authorities intervened. Even then, Mary wouldn't give in, and their attempt to capture her culminated in a 3-hour foot chase. Mary was apparently pretty fast on her feet. Despite an all-out effort to avoid it, Malin was finally apprehended and taken into custody in 1907. Doctors took urine, stool, and tissue samples, which eventually confirmed that Malin was carrying the typhoid bacteria. This, despite the fact that she displayed no symptoms herself. By all accounts, she seemed to be a picture of health. With no cure available, health officials had to find another course of action to protect the public. Malin was deemed a threat to society, and it was decided that she would be quarantined. Though she fought every step of the way, she was eventually confined to a single occupancy cottage at Riverside Hospital for Communicable Diseases, located on New York's North Brother Island. When confronted by reporters, Malin said she believed she was being treated unjustly, and she continued to insist that there was no way she could have typhoid. Nonetheless, Mary's case attracted so much attention, she was labeled Typhoid Mary in a 1908 issue of the Journal of the American Medical Association, and the name stuck. Mary Malin wasn't America's first typhoid carrier, but she was the first asymptomatic carrier ever identified. This means that she was able to carry and spread the disease without ever showing any of its symptoms herself. That sounds familiar. This was considered a hugely important scientific discovery. But that being the case, there was no existing protocol that addressed the situation. While the authorities did have the power to forcibly quarantine anyone who posed a threat to public health, and they knew Mary was somehow mixed up in all the typhoid outbreaks, they couldn't prove how or why she was involved. Mary didn't take kindly to the attention. She angrily claimed to be a victim of a government conspiracy, and at some points the public even seemed to agree with her. Regardless, authorities knew she had to be the carrier and were desperate to stop the trail of sickness and death she left in her wake. Scientists of Malin's time were never able to figure out how she could carry the bacteria that caused typhoid without showing symptoms. But in 2013, a group of researchers at Stanford solved the riddle. To put it as simply as possible, the Salmonella bacteria behind the spread of typhoid has the ability to hide in immune cells known as macrophages and hijack their metabolism for their own purposes. If this hijacking is successful, the person in question can spread the bacteria while themselves appearing healthy. In 1910, Mary Malin was released from her first quarantine under the condition that she would never again work as a cook. She took a job doing laundry, but it didn't last long. She left the position after a short time and returned to cooking for families. The authorities got back on Mary's trail, but she managed to avoid them by frequently changing jobs and names. Finally, in 1915, Sloan Maternity Hospital in New York had an outbreak of 25 new typhoid cases. Malin was discovered working there as a cook and was quickly arrested and returned to quarantine. By 1915, Malin was infamously known to all as Typhoid Mary. She had been recaptured and placed back in quarantine at North Brother Island in New York. Her life there was not pleasant. Whether due to her intransigence with the doctors and staff or the facility's complete inexperience in handling cases like Mary's, it's known that she was treated inhumanely. The initial tests performed discovered that Malin's gallbladder was riddled with Salmonella bacteria. They wanted to remove the organ during her first quarantine, but she refused to allow it. They tried again during her second quarantine, and once again Mary managed to avoid the procedure. She wasn't able to avoid everything, though. Over her remaining years at the facility, doctors took over 160 biological samples from her body, all against her wishes. As if that wasn't indignity enough, Malin was often shown off to journalists and interns as a specimen. When she wasn't being prodded or exploited, she was being neglected. As her interactions were severely limited by the doctors, she was only allowed to wash bottles in the laboratory. Dubbed the most dangerous woman in America, Mary Malin had finally worn out whatever goodwill she had left with the authorities. Deciding that she simply could not be trusted to give up cooking or follow any sort of prevention guidelines, New York State public health officials decided she had to be quarantined for the remainder of her life. That's right, for life. She spent her remaining years in virtual isolation in the quarantine cottage on North Brother Island. Mary died in 1938, and though reports of the matter conflict, it is believed she likely passed away from pneumonia. It's not known how many cases of typhoid she spread in her lifetime, but estimates suggest she caused no less than 51 cases, resulting in 3 fatalities. While the discovery of an asymptomatic typhoid carrier was big news in 1906, by the time Mary died in 1938, it was old hat. By that point, over 400 other healthy carriers had been identified. Yet, according to the surviving records, not a single one of these others received the same forced confinement that Mary Malin did. The jarring discrepancy raised important philosophical questions about epidemic and public health protocols. Some began to wonder whether forcibly confining someone for the greater good of society at large was justifiable. Others pointed out that it was Mary's lack of honesty and refusal to cooperate that merited such severe treatment. These issues are still relevant in modern epidemiology. So what do you think? Do you feel any sympathy for typhoid?
Okay, so that's it. In the video, and you watched the sad story of Mary Malin, and there was a time when people started just right at that point started to know about germs. The people didn't have knowledge. To the extent we have today. But even now, if I'm just trying to draw a kind of a parallel here, when coronavirus is affecting kind of global pandemic, it's being caused here, and we see some kind of ill treatment, inhuman treatment to the people or to the family which have members with this coronavirus affected or asymptomatic carrier, whoever, whichever way, contracting diseases. And there are situations which shows humanity, but many other situations we see here and there where we are behaving very inhuman, not knowing anything scientific, and trying to seclude them in a completely separate zone, and they should be avoided for life or something like that. They should not be talked to, or they should not be someone who we can come close even after they have been cured. So this kind of situations, today we need to be careful so that we don't repeat the same kind of situation that happened to Mary Malin and many other people who were there as asymptomatic carriers. So that's all. I believe you have got at least a little bit of idea, even though the entire talk was being very much troubled. At least I felt enormous trouble dealing with this mouse and keyboard and all this stuff. Nothing has been controlled, and now it seems that it's all working. Nothing is moving without my order when I'm about to end the talk. Okay, so before I conclude today's webinar, which is the last one of the 4-day series of webinars on medical biotechnology and bioinformatics from the perspective of research and career prospects, I would like to invite you for the upcoming ones from a different center of our own institute, JIS Institute of Advanced Studies and Research. The center is, it's called the Center for Interdisciplinary Sciences. They're having a 2-day webinar. The first one will be held tomorrow at 3 o'clock, and the next one on Monday, that is August 10, same time, 3 o'clock. And the topic is Sustainable Materials, Polymers, and Sensors. Tomorrow's talk will be offered by Dr. Prashanjit Saha, who got his PhD from IIT Kharagpur and presently an assistant professor in the Center for Interdisciplinary Sciences of JIS IASR. His topic is going to be polymer science and technology and 3D printing. And on Monday, Dr. Shubham Kashingho, he got his PhD from Postek, South Korea, and also an assistant professor in the same center. He's going to talk about materials for bioimaging, chemo, and biosensors. So you please feel free to go to Facebook page of JIS IASR, where you can get a clickable link through which you can register yourself and join these two exciting, insightful presentations tomorrow and on Monday. And finally, again, as we have talked again and again about the importance of bioinformatics, importance of medical biotechnology in health and medicine throughout the era of science, and it is even more now, especially from the perspective of infectious diseases. Admissions are open for the 2-year courses of MTech and MSc, and we are taking PhD interested students for doing PhD. You can do regular PhD, or you can have you can pursue integrated program, master's PhD program. You can contact us in any of these 2 numbers, or can email us at biosynth@jisiasr.org, and also at admission@jisiasr.org. And you can also go to our webpage, jisisr.org, and there you will have this one tab called Admissions 2020, where you can click and apply online. And these are some of the factors that are already shown. You might have missed, video presentation was awful today, but somehow I believe you got the message. So we, as a newborn institute, yet to complete our first year, we have achieved a lot compared to any other government or private institutes here in India. I mean, I'm talking about India, not just the Eastern Zone. In the first year, in course of research projects and all these things, we have got extensively kind of high quality faculty pool, and the resources, the lab resources, we have got all these things. And we offer scholarships to meritorious students. That is one thing we want to build. We want to develop ourselves together with the students. Without meritorious, motivated students, it's totally impossible for us to grow. That's why we're looking for very high quality students. So if you think that this is something which you want to pursue, but you cannot afford, definitely come to us. We will make it happen in one way or the other if you are meritorious enough. Otherwise, definitely it's an exciting career ahead. I'm telling you, if you want to pursue courses, whether you want to go to industry or, R&D of industry, or to any academic institutions here or abroad. So that's all from me. Let's see if you have any questions. I'm happy to take questions.
Thank you, Professor Chattopadhyay. It was really a splendid talk, and the last 2 hours were so much interesting for us. We got to know so many things about microbial evolution, starting from very beginning. So now it's time for the questionnaire session. If you have any question, please write in the question and answer section. Professor Chattopadhyay will happy to answer your questions. And I can see one question from Jonathan Shen to you. In some cases, without changes of gene sequences, there are some minute changes in protein or enzyme function structure, and which leads to evolutionary changes. How can we determine with bioinformatics tool?
Minor changes in the function, minor changes in the function without any change in the structure, meaning without any change in the sequences. Yeah, definitely. Then this is definitely not part of this computational genomics. So then it goes to the area of proteomics. Then you need to go for some kind of proteomic analysis, or you have to start. I mean, experimental analysis is something, but if you want to go for bioinformatic tool, it will ultimately depend on the variations. If you have the same sequence, then it is, I mean, it's not going to give you any such result. I mean, you can do for functional perspective, you can do mass spec and some other NMR study and some other kind of analysis. But as far as the bioinformatic tools are there, unless and until there is any change in the amino acid sequence or in the gene or in DNA sequence, it's, I mean, it is outside of the role of this bioinformatic tool, actually, so far I know.
There more attendees are there who are thanking Professor Chattopadhyay for such a great presentation. And then another question is there: for studying convergent sequence difference for bacterial 16S gene, what is the free software to be used?
Yeah. Can you repeat the question again?
For yes, of course. For studying convergent sequence difference for bacterial 16S gene, what is the free software to be used?
Bacterial convergence sequence, I mean, you can, I mean, there are several software, especially for convergent evolution. There is no automated software for non-protein coding genes. So for that, you have to go, there are tools like DNA-SP, DNA sequence polymorphism, DNA-SP, or you can use MEGA to map the evolution, map the mutations, and there you can find out whether the changes are converging, whether you find convergent changes, but that you have to do manually. This analysis of convergent mutations not yet incorporated in MEGA. It could be someday in future, but at this point, it's not incorporated in any tool. But if you want to go for the analysis of convergent mutations in protein coding genes, then you can use a software called Time Zone, TIME, Time Zone. That is one software that's kind of deal with this kind of convergent evolution of protein coding genes. For non-protein coding genes, you can definitely map the mutations, and then you can manually find out whether there is any convergent, meaning that they are occurring, they're emerging, those mutations are emerging independent of one another.
Thank you, sir. The next one from anonymous attendee to you is, sir, changes in amino acid sequence can change the protein function? Is both are related?
Yes and no. The thing is, the change in amino acid sequence can lead to any of the following possibilities. One is that there could be change in protein function that can be helpful for the organism, that might not be offering any change to the organism, or that could actually be deleterious, or a change could also make the protein non-functional. There are all different possibilities. A change in the amino acid sequence can lead to no change at all. That is one possibility. Can lead to the inactivation of protein, that is the loss of function. And the third scenario is definitely what you are saying, that it can change the protein function. And then that change can be the increase in function or decrease in function. All possibilities is there. Or it could be neofunctionalization. It could be the protein could be offering a completely different function. So three possibilities are there if you relate amino acid sequence change to protein function. But there are additionally two other possibilities where the protein can lose the function and there could be no change at all in the protein function.
The next question is again from an anonymous attendee. Can protein function be predicted from sequence?
Well, protein function prediction, definitely first thing is to predict the structure, and then definitely you can predict the function to some extent using some additional information. Just by having the sequence, you cannot predict the function. You can predict the structure, and then if you have known function of homologous structures or some other proteins which are having the same structures, so you can use that information to predict the structure of the protein of your interest. That is how you can go. But I believe we have our colleague Dr. Devabani Ganguly, who presented the webinar on the very first day from Newton's law to drug design. There, I believe she dealt with this kind of issues. And if you want any kind of help, you can directly write to us at biosimp@jisiasr.org, and we'll be happy to help. But this is the normal scenario. I mean, without having any additional information, it's unlikely that from the sequence, I mean, you have to have some information from other databases about the structure, about the function of similar structures or similar sequences. And that is the way you can extrapolate that information here. But that would be very I would say, not mature information. You have to do, finally, you have to go for experimental analysis to perform and then talk about function. But definitely, depending on the metadata or depending on the additional information you have regarding similar structures, you can strengthen your predictions.
The next question is, sir, do you work on Salmonella MLST, housekeeping genes? What software is used to study MLST?
First of all, studying MLST, I mean, I believe if you work on MLST, there is one big software, big database, which originally was created by Mark Hoffman and his group in Germany, but now I believe it's hosted in Ireland and somewhere else. It's called PubMLST. You can use that, and if you have sequences of your Salmonella or whichever organism you want to work on, you can sequences of the MLST loci, the loci or the gene fragments that are used as MLST markers for that particular species. If you have those sequences, you can submit those sequences in PubMLST, and you can get the information about the allelic profiles. You can, once you have all the allelic profiles, you can get the information of the sequence type. That is what is called ST in short. And from there, you can have the information of the clone and clonal complexes of the species of your interest, maybe Salmonella or anything. I personally definitely work on, I mean, MLST is something that we always do, even if we have the whole genome sequences, we always start with MLST to identify the clonal structure, the clonal diversity of our samples that we are going to analyze. Definitely we do that. And software-wise, we have quite a few in-house tools, but there are, I mean, many, as I'm talking about, like when you do this alignment and all these things of this MLST loci or any other gene sequences, I would suggest you to use Mega. Mega is built in US. It's by Sudhir Kumar, an Indian scientist. So it's very rich in functionality, very rich in features, very powerful tool. You can use Mega and you can use DNASP, and many other similar tools are there. And you can also use, we normally either use Mega or use our in-house tools, depending on our needs. What you want to do, what we want to do on the MLST thing, what kind of analysis, whether we want to concatenate the MLST loci and create a single MLST sequence for a given organism or something like that. Depending on the need, we choose the software.
It was really a brilliant presentation. So the question is answer section, and the chat is flooded with the thank you from your attendees, Professor Chattopadhyay. And I got a question from Bilkis Rahman, which is certainly not for you, sir. So for all the organizers, she has asked that since it's the last day of this series, when are we going to get the e-certificate? Regarding this, I can say that we will notify you once such a decision is made, and you will get it by the same mail you have been notified for this webinar. And again, if you have any questions for Professor Sujoy Chattopadhyay, already we have given you the mail ID biosimp@jisisr.org. Kindly put all the questions over there. He will answer your mail definitely.
Just one thing I would like to mention. I would like to apologize to the attendees, and especially to ones who suggested, many people, I am now looking at the chat section, and many people suggested a way out, solution to the problem I was facing, and then I was such an idiot that I didn't look at the chat section, and that you are all out to help me there, and then I just ignored those things. I didn't look at the chat section that they're saying the PowerPoint slide timer is switched on somehow. And then, yeah, so please change the slide timer. So thank you so much. Shubham Dash, Niranjan Pal Chaudhuri, and whoever else helped me or tried to help me. I didn't take, that's my misfortune. Sorry about this disturbance, and sorry for missing your kind of help to resolve the issues.
And another attendee is there who has told you that, sir, would you like to deliver a lecture at our institute? Please send me your email and number. So I would like to say that you can drop an email to the biosimp@jisisr.org, or in our website you can get all the details about Professor Sujoy Chattopadhyay under the division of Center for Health Science and Technology. So from there you can get all of his details and contact him.
And also you can get it here. sujayc@jisr.org. I believe if you are still there, you should be able to find the email address from this slide. Is the slide visible, Dr. Banerjee?
Yes, it's visible.
Okay. Thank you. Thank you so much.
Thank you to all the attendees, and do not miss the webinars conducted by our Center for Interdisciplinary Sciences on tomorrow and Monday, that is August 10. And we will like to have all of you as our attendees there also. Thank you.